Here you have a comparison of the speed of the different types of muscle tissue. Fastest one being here skeletal muscle, and then cardiac muscle and smooth muscle. You can tell it's really smooth, sustained kind of contraction, these peristaltic wave-like contractions, so much slower. And take a look at the timeline here, right here. Compare that to the action potentials. That's when we had milliseconds. Now here we have full seconds on the timeline. So overall, muscles are a lot slower than like nerve impulse conduction. But overall skeletal muscle pretty fast though, cardiac muscle a little slower, but then smooth muscle really slow and kind of wave-like. Okay, so let's take a look at how smooth muscle works. It is not arranged in the typical type of sarcomeres that you would see in skeletal muscle and even in cardiac muscle. The contraction is still initiated by an electrical or chemical signal or both. And definitely under the control of the autonomic nervous system. Don't ever forget, it's one of the effectors of the autonomic nervous system. We had smooth muscle, cardiac muscle, and glands there. And we don't have specialized receptor regions there, but we're running these varicosities through the whole tissue and then dumping neurotransmitter onto the tissue. You need the calcium is from the extracellular fluid and from the sarcoplasmic reticulum. And the calcium initiates a cascade ending with phosphorylation of myosin light chain and activation of myosin ATPase. And we'll take a look at that. And smooth muscle overall, the protein actin is has it's a larger percentage of the overall muscle protein. It's a different type of actin too. It's called smooth muscle actin. We don't have troponin, but you will see that we have a replacement that kind of is similar. We don't have as much myosin. The myosin filaments are a little bit longer, and the entire surface of the filaments covered with the myosin heads. We do have an extensive cytoskeleton in these smooth muscle cells, and we have the sarcoplasmic reticulum amount varies, is less organized. It's not like those triads that we have in skeletal muscle. We don't have T-tubules. We don't really need them. We don't have these extensive, large muscle fibers, but instead we have Cameo Lime. And here's what it looks like. So it does kind of resemble sort of a sarcomere and skeletal muscle, but then you do have the smooth muscle actin right here. And in this case, the arrangement here is we're moving in opposite direction. So this actin filament here moves in this direction, and this upper row here moves in the opposite direction. And then here, kind of similar, the myosin filaments, the stem portions are kind of strung together as we saw in skeletal muscle, and then the myosin heads are sticking up, but again sort of we're moving in opposite direction here rather than in the same direction. And let's take a look at sort of the intermediate filaments, these dense bodies that are forming. Just kind of pay attention here to the actin and myosin arrangement. It kind of resembles sort of the sarcomere structure with the myosin in the center right here, still in purple. And then instead of having more this box shape surrounding, it's a little bit more rounded, but still somewhat resembles the sarcomeres that you would see in skeletal muscle. And basically the arrangement here, you have these dense bodies that are sort of anchoring everything to the cytoskeleton too. So here is a summary of what happens, and I'm going to walk you through this step by step. You can go back to the summary slide later and sort of put it all back together, but here let's sort of go step by step. We still need calcium as sort of the initiator of the muscle contraction. But instead of binding to troponin as a sort of regulatory protein, we're going to bind the calcium this time to calmodulin. Calmodulin is, in a sense, it's kind of similar to troponin. It has high affinity to calcium, and it changes its shape when it's bound to calcium. So in that sense, it's kind of similar, the troponin into calmodulin. So here in smooth muscle, we are binding the calcium. Once we have the calcium released here from the sarcoplasmic reticulum and also influx here from the extracellular fluid, the calcium will bind to calmodulin, making it calcium calmodulin. The calcium calmodulin will activate a protein, an enzyme that's called myosin light chain kinase. And so it will switch it from an inactive to an active myosin light chain kinase, MLCK. A kinase, as you know, phosphorylates, and in this case, well, we're phosphorylating the myosin. That's what the enzyme does. That's what the name says, myosin light chain kinase. So we are phosphorylating the light chain of the myosin molecules right here, and that basically powers the heads. And then we will get a sort of a resemblance of this myosin-actin cross-bridge cycling, kind of similar to what we saw in skeletal muscle, but here in steps. So we will have the influx of calcium from the extracellular fluid and from the internal stores here from the sarcoplasmic reticulum. The calcium will bind to calmodulin, and that will make it calcium calmodulin. And calcium calmodulin will activate an enzyme that's known as myosin light chain kinase. Once the myosin light chain kinase is activated, it phosphorylates the light chains on the myosin, thereby activating it. And once the myosin here is activated, then you will get the cross-bridge cycling of actin and myosin. Let's take a look at how we can stop that contraction, and for that we would have to first pump the calcium outside of the cell back into the extracellular fluid compartment or back into the sarcoplasmic reticulum. Once the calcium concentration is lowered and the calcium comes off of the calmodulin, and we're gonna have just calmodulin without the calcium, that means that the myosin light chain kinase activity is going down, and instead we're going to activate the myosin phosphatase, which removes the phosphates from the myosin heads there, and that inactivates the myosin heads, and you no longer have the interaction of actin and myosin, and thereby you get decreased muscle tension. So in steps here, first thing we're going to have to do is remove the calcium. So we're going to pump it outside of the muscle cell, the smooth muscle cell that is, and we're going to put it back into the sarcoplasmic reticulum. And once the concentration of calcium is lowered, then the calcium will come off of the calmodulin. So here then if we have just calmodulin without the calcium, and that actually will decrease the activity of the myosin light chain kinase, and then we are going to use a myosin phosphatase to remove the phosphates off of these myosin heads. And once that has happened, then we're going to have decreased interaction of actin and myosin, and that means the muscle relaxes and any kind of contraction, tension development will stop. So here you have everything in summary in one slide. Again, the removal of calcium from the inside of the cell starts it all out to get the relaxation going. We need to remove the calcium from the calmodulin, and then we need to inactivate the myosin light chain kinase. And instead we need to activate a phosphatase that removes the phosphates of the myosin heads, and that's how you end up with your relaxation. Now, so the dephosphorylated myosin could remain attached for a certain period of time in this smooth muscle contraction, these peristaltic kind of contractions. And the contraction itself is caused by electrical signaling and electrochemical coupling. Then there's also something called pharmacomechanical coupling that I don't want to spend much time on, but let's take a look at the slow wave-like contractions that we see in smooth muscle. So here would be subthreshold waviness going on, but you have to reach threshold before actual potentials will be fired. And here's another example of the pacemaker potentials. They will always depolarize the threshold. So you have typically some additional ion channels here. So you see that we don't spend much time at the resting membrane potential. So these these cells typically have an unstable resting membrane potential, and they will reach threshold at a certain rate automatically, and that's why they're called pacemakers, because they automatically will go back to threshold, fire an action potential, repolarize, stay down there for a little bit, and then fire again. So we will see that in the heart, and that we have at the SA node, we have also pacemaker potentials. Again, this pharmacomechanical coupling, I don't want to spend much time on this, so we should move on here. The calcium release from the sarcoplasmic reticulum, there are these ryanodin receptor calcium release channels. That's greetings from skeletal muscles. Also where we had the ryanodin receptor, that's the gatekeeper in the sarcoplasmic reticulum. We also have this calcium-induced calcium release, and you don't have to worry about that too much. For the most part, we're dealing with IP3 receptor channels. That's the most common pathway, that you have IP3, that's second messenger that we talked about in, what was it, chapter 6, something like that. IP3 is a second messenger that opens calcium gates. It's notorious for doing that, and it will release calcium then from stores, internal stores. The cell membrane calcium entry, that's operated through voltage-gated channels. They're not voltage-graded, I'm sorry. They're voltage-gated. Just take that R off. That's silly. Voltage-gated calcium channels. Then we also have some ligand-gated calcium channels or receptor-operated calcium channels. I don't need to worry about it too much. There's also something called stretch-activated calcium channels. They open when pressure or other force distorts the cell membrane. That would be known as a myogenic contraction. And then here's some sort of the summary of all the different ways that you could control smooth muscle contraction and how you can get that calcium release. I would say probably this here is sort of the most important part here, the IP3 that is opening calcium gates, and that's how you get the calcium release, and that's how you can then bind the calcium to the calmodulin, and then you get your muscle contraction going. So you don't need to pay too much attention to these different specific pathways. And smooth muscle regulation here, sort of finishing up on this lecture. As you know, smooth muscle is an effector of the autonomic nervous system, and a lot of times you have both sympathetic and parasympathetic neurons that are innervating these smooth muscle tissues. You can also have hormones and paracrines, or locally acting messenger molecules that control smooth muscle contraction. Histamine, for example, is a substance that's releasing allergic reactions, a lot of times constricting the smooth muscle of the airways, and that's what you have when somebody has an asthma attack, an allergic reaction to something, and it feels like that something is just almost choking them. So that's the smooth muscle constriction of the airways. Nitric oxide is known for relaxing smooth muscle in blood vessels, so the lining of blood vessels. So that is a known vasodilator. Nitric oxide, very powerful vasodilator. And a few words about cardiac muscle, but we have a whole lecture on cardiac muscle and how it operates. It's actually very similar to skeletal muscle. It does have striations, just like skeletal muscle. It has this typical sarcomere structure that we saw in skeletal muscle. But of course, it's not under your voluntary control. It's controlled by the autonomic nervous system, one of those effectors of the autonomic nervous system. Also, another thing that's different from skeletal muscle is that the fibers are shorter. They may be branched. They have a single nucleus, indicating there was a single cell that they originated from. But then they also have these intercalated discs that connect one cell to the next. Here, continuing, cardiac muscle is like smooth muscle because it's electrically linking the cells to one another. Some have pacemaker potentials, which you will see in the heart lecture. And then we, of course, have the sympathetic and parasympathetic control, also some hormone control. And then again, in cardiac muscle, the feature, we have the gap junctions and the intercalated discs. Now, our very last slide right here, that is a summary slide. And here we're comparing the 3 different types of muscle: skeletal muscle, smooth muscle, cardiac muscle. So you should take a look at this. This is a nice summary table that summarizes the features of the 3 different types of muscle that we talked about in this lecture. Pay attention to the fiber arrangement, locations, morphology. You can take a look at that. Internal structure, how it is controlled. Of course, that is very important for physiology. And then some of the other features that you will see in skeletal versus smooth versus cardiac muscle. So take a look at that and study from here. And don't forget to look at the video clips that I posted.